Hi, my name is Troy McElking Jr. I was born in New Orleans, Louisiana, and raised in Powder Springs, Georgia. I went to college at Oklahoma State University in Stillwater, Oklahoma, and I graduated with a dual degree in biochemistry and microbiology in the spring of 2016. My interest in science began as early as elementary school, but more particularly during my 12th grade biology class. During this class, my teacher presented science as a fascinating prospect, filled with vast mysteries within our genetic code and the organisms we interacted with. During my first year at Oklahoma State, I wanted more exposure to science. While researching science degrees, I found that advanced careers in science required an education beyond a bachelor's. For my ambition to conduct research, I was allotted my first opportunity by a graduate student when I expressed interest in aiding him with his project. This experience further rooted my admiration for science, and I continued to grow and evolve over my additional research opportunities. My current research interests involve the study of host-pathogen interactions. I am particularly interested in understanding the mechanisms pathogens possess to evade host immune responses and develop infection. When looking at long-term prospects, I envision myself as a research professor at an institution where I can mentor students to pursue higher education. Research, to me, is an outlet for creativity and wonder. As a professor, I aspire to become a role model and inspire future students to share my passion for science. I joined PREP because I wanted an opportunity to conduct research in an infectious disease lab where I can explore host pathogen interactions. I also wanted to obtain additional laboratory and critical thinking skills that will prepare me for future graduate studies. PREP at UGA has been a program that focuses on my desire to grow in my writing and oral communication. I love PREP at UGA's connection with the Department of Infectious Diseases because this is a field that most fascinates me. Through PREP at UGA, I have resources such as an individual development plan and presentation feedback forums. These align with my desire to become more proficient in science and achieve my goal to be a principal investigator. There are several skills I plan to acquire from my time in PREP at UGA. First, I hope to increase my scientific literacy to be able to express my ideas to a broad range of individuals. Second, I plan to build a strong foundation for becoming a disciplined and passionate scientist so that I can motivate others to consider a career in an exciting field of science. Lastly, I want to increase my confidence to be an independent component of a lab to form ideas for experiments that exhibit advancement in the field. My faculty mentor is Dr. Eric Harville, and my research mentors are Dr. Holly Vuong and Dr. Ilyasu Hamadou Samana. Together, we are addressing questions on the interactions of pathogens and commensal bacteria. My research project is to understand the role of the respiratory microbiota in protecting the host against an invading pathogen. We have two Bordetella species that produce very different results in how they interact with the host microbiota, even though they are genetically very closely related to each other. Bordetella bronchoseptica and Bordetella pertussis are the two species we research. Dr. Harville's lab has shown that pertussis, the human pathogen, cannot easily invade into a mouse, unless that mouse was given high amounts of the inoculum. However, bronchoseptica very easily infects mice with very low amounts. It appears that bronchoseptica can disrupt the nasal microbiota and occupy the nasal cavity, and still be the primary culpable bacteria even after 70 days of the initial infection. I hypothesize that host specificity by Bordetella species can affect their disruption ability and colonization rates in hosts. Because we have such a good mouse model, my work will focus on how bronchoseptica displaces the nasal microbiota. To get the larger picture first, we need to carry out simple, clearly defined competitive experiments in cultures. Then, we will study their interactions and understand the mechanisms behind the disruption before we can move to the actual mouse host. My approach to answering this question involves breaking down the composition of the mouse nasal cavity using 16S rRNA sequencing to identify potential candidates for interaction with bronchoseptica. I will identify the specific conditions to study clearing in culture. I will also strengthen the growth assay to determine important genes for this clearing mechanism by using mutant bronchoseptica strains which lack the clearing mechanism found within the mice. In order to further test implications of pertussis clearing in human microbiota, I will manipulate mouse microbiota to acquire the appropriate conditions to test pertussis colonization strategy. The Harvard lab has been extremely supportive in helping me develop and expand upon my hypotheses. The collaborative process during this project has been crucial for my development as a scientific researcher. My name is Troy McElking Jr., and I'm a PrEP at UGA scholar.